So this data then is matched up with the mapping software that comes from Genome Studio of where each SNP is on the chip, and then that's imported into our Illumina Genome Studio software. So you have to have this software to work with the Infinium chip. And just for people that aren't familiar with it, what we're looking at here is a screenshot from that. And I have the cursor along one of the rows, rows of our SNPs. So this column here is our various SNPs, and we have them sorted in some order. But the key values that I want to point out here is the theta value and then the R value here. And those two values are used to graph the SNPs onto this graph here. And over here, which you can see is actually the progeny or the samples are listed, and we can kind of color highlight different samples within this graph for ease in viewing. So now the Illumina system, the Illumina genome software has auto calling for the SNPs. However, in the SOLCAP project, what we did was we actually created a custom potato calling, and for the 3 clusters, of the 2 homozygous classes and the heterozygous class, and we based this upon our potato diversity panel, 2 tetraploid populations, and 1 diploid population. And so this custom calling file can be accessed at our SOLCAP website. So of the 8,303, it was determined that 7,412 of those SNPs were considered good for 3 cluster calling. So if we're looking now closer at those Illumina Infinium graphs of our SNPs, what we're looking at here is one SNP example, and what we have here is one parent in the homozygous AA state, and we have one parent in the homozygous BB state, and the progeny from that should be expected to be all heterozygous AB, and that's what we see here. On the second graph in this slide, you can see we have one parent is a heterozygote AB and the other one's a homozygote BB, and when we cross them, we have a 1-to-1 distribution in the progeny for the heterozygote and homozygous classes. So it's a very easy system to read, and what we're seeing along the x-axis here is the theta values. So where the theta value is kind of helps us position the call on the SNP, and the R value I think is a reflection of the intensity of the signal that the system was reading. Okay, so we're looking at it now we've looked at it from a diploid level. So what we really need to do is start thinking about tetraploids, and so that can get some people's heads swirling, and or lead to some nightmares. But when you have tetraploids, you have a different type of segregation patterns than what we normally see at the diploids. And so we can have 1-to-1 segregations, 1 to 4-to-1 segregations, 1 to 2-to-1, 1 to 5 to 5-to-1, and then the most complex is the 1 to 8 to 18 to 8-to-1. Okay? So what's happening in each of these tetraploid segregations, we have up to 5 different genotypic classes. And so the Illumina Genome Studio software is designed for calling 3 clusters, and what we need to be able to do is call 5 clusters so that we can differentiate between the 3 heterozygous groups, which we refer to as simplex, duplex, and nulliplex, which are the different doses, in this case, of the B allele, of a single dose, double dose, and a triple dose. What's really been exciting about this Infinium platform is that the SNPs are codominant, and many of them are dosage sensitive, and so we really do have a very ideal marker to look at our tetraploid populations. So as a next step, the same panel of potato lines, the diversity panel, the 2 tetraploid populations and the 1 diploid population, were then looked at to develop a 5-cluster custom calling based upon the theta values. So each SNP was looked at, and there was a lot of effort put into this, and as a result of that, it was determined based upon our data set that we have a little over 5,000 SNPs that we can use the 5-cluster custom calling. And we had about 3,272 that there was only 1 cluster or was a bad SNP. So now we're looking at our tetraploid populations here on the Illumina Genome Studio graph, and we have here, we can see we have parent 1, parent 2. On the left side we have a diploid population, and again we have the heterozygotes all clustering to the middle of the graph. On the right hand side we have a tetraploid population. We have a cross between a duplex heterozygote and another homozygous parent. Oh no, sorry, take that back. We have a cross between our simplex heterozygote and our duplex, and so you can see then the segregation between the 2 genotype classes there. And so we consider this ideal because it works both for the diploid and the tetraploid germplasm. So now what we're going to do is look at some of the data that actually came off our Premier Russet by Rio Grande Russet mapping population. So as you saw earlier, there was 184 progeny that we're basing this mapping population on. That number is the number of progeny where we have both phenotype data and genotype data in a usable form, and to do the analysis. So looking at this first graph here on the left-hand side, what we have is a cross between Premier Russet being a simplex heterozygote to Rio Grande Russet being a nulliplex. And so what we expect is a 1-to-1 segregation between the nulliplex and simplex classes. And you can see the data up here, and we thought it was quite cute that the numbers came out exactly. We are not trying to be like Gregor Mendel and make our numbers fit, just for people that are questioning things. And oops, sorry, I jumped ahead by accident. And then on the right-hand side, we now have a cross between our nulliplex for Premier Russet and a duplex SNP in Rio Grande. And here we expect a 1-to-4-to-1 segregation ratio, and here you can see the numbers that we obtained in our progeny, and those fit that expected ratio. Now just one thing to point out here is on our screen here we're seeing the circles drawn for the auto-calling in Genome Studio, whereas what we're doing is actually basing our SNP calls on the 5-cluster custom calling. So those circles are kind of something that's difficult to remove from the graph at this point. Okay, so now looking at a different segregation pattern, what we have here is a duplex heterozygote cross to a simplex heterozygote. And that's going to give us that 1-to-5 to 5-to-1 segregation ratio. So we have 4 segregation classes that we're scoring in this, or 4 genotypes that we're scoring in this progeny. And so you can see again we've got the separation into those our heterozygotes into the 3 heterozygous genotypes. And then lastly, what we have is that complex duplex by duplex segregation, which gives us the 5 genotypic classes and segregating in that 1-to-8 to 18 to 8-to-1. Now, as we were looking at the data, we kind of faced a reality point, and one of the questions they always had is, well, how good is the SNP calling? You know, are things going to be miscalled or not? And when we started looking at the data, so in a simplex, nullplex by simplex, as I showed earlier, we expect a one-to-one segregation. However, because potatoes are tetraploids and have autotetraploid type segregation, one of the consequences of that is we can actually see products of double reduction. And hopefully my potato colleagues are familiar with this term. And so this brought back some fond memories of my earlier days of looking at double reduction. And so what we are seeing double reduction products with the SNPs. So what we have here is we expect the one-to-one segregation between for the nullplex and simplex, but a double reduction product in this cross would be a duplex, and we can see them clustering separate from the simplex. And we have on the right-hand side we have another example of that in a cross in the other direction where we have a B homozygote to a B to a ABBB simplex or triplex. So so we were seeing this a number of times. And so if you go back to your cytogenetics, what you can see is that the double reduction is going to happen when you have autotetraploid pairing in meiosis. And if you're really interested in this subject, you can go to this book by Burnham, Discussions in Cytogenetics, 1962. But just as for you to walk you through this, is that double reduction occurs when the two sister chromatids are recovered in a single gamete. And what this requires is multivalent pairing, and then a crossover between the locus and the centromere, and then the two pairs of the chromatids passing to the same pole. And so if you were to work out the probabilities of this, it happens about that in a simplex cross, we're going to see that approximately as a maximum of 1/24 of the time. So, but it's the frequency of the double reduction is proportional to how far it is from the centromere. So loci that are closer to the centromere, we're not going to see that. And so this table here, we graphed out all 800 simplex segregation patterns that we saw in our SNPs in this population. And so 541 of the SNPs, we did not see any double reduction products. And as you can go down the list, you can see as the number of double reduction products goes up, the number of SNPs that had those number of double reductions goes down, because the SNPs would have to be far from the centromere for us to see those. Okay. Using some graphical software in the statistical program JMP, Joe Coombs was able to plot this out. So what we're looking at here is we've got our 12 chromosomes, 1, 2, 3, and so on. And what we have across is the linear order of our chromosomes in megabases. Okay, so what we'd expect to see, and I think it's seen clearly here on chromosome 4, is that our double reduction products would tend to be more frequent at the ends of the chromosomes where they're far from the centromere. So you see this clearly in chromosome 4 and in chromosome 5 and 6 and so on. This we haven't really delved into this data much, but this is what we're seeing so far, and so I just wanted to pass it on, because we have double reduction is something that's happening, and we got to be aware of it. And if you're using the tetraploid map software, it actually does not handle double reduction products, so those have to be removed from the analysis.